So my name is Snir. I'm a senior product manager at MyHeritage. I'm handling our DNA product. So pretty much what you get to experience on the website or on the app as a result of what my team and I are doing. In this session, I will talk a little bit about the basics. We will talk about basic terms, glossary, related to genetic testing, genetic genealogy, DNA. And I'll also explain how are we able to come up with the results that you eventually see on the website. Some think it's very easy. You take a test, you send it, and that's it. But there's a lot more into it. So as I mentioned, we'll start with the DNA at a glance, and then later on talk about the actual science behind what we're doing with the raw DNA data results. How many here have tested for DNA? Woo, very nice. Thank you so much. So, if you've taken a DNA test with us or have uploaded from another company, you're probably familiar with these two screens. On the left, you can see your ethnicity estimate, and on the right, you can see your list of DNA matches. But that's not really what we know about you after you take the test. There's a lot much into it. So this is my match with my dad, Rafael. And on the right, you can see DNA match quality. And if I'll zoom in for a second so that it's a little bit bigger, you can see the amount of shared DNA, how many segments, what is the largest segment, these two letters CM, which represents Centimorgan. This is not the height of your DNA match. Do not be confused. So this is kind of like the raw data, the basic information that we have that we can tell you about this person. But let's try and understand first, what does it mean? So DNA at a glance, all of us, human beings, we have 22 what we call autosomal chromosomes, and an additional chromosome, which is the sex chromosome that defines our sex, an XY for a male, and two X's for a female. And this is pretty much how it looks for all of us. This is our combination, our genetic structure. But it's not that different always. So here we have two genomes on the top and on the bottom. And these I took from two different individuals. Anyone sees there are some minor differences. Anyone has a guess? What's the difference between the one on the top and the one on the bottom? Because they look pretty much the same. So the truth is that above we have a human, and at the bottom we have a chimpanzee. So we're not that different. And actually there's a 98% similarity when we look at the DNA of a chimpanzee and a human being. They look 98% exactly the same. When it comes to human beings, things are getting even more close, and it's about 99.9%. Similarity in the genetic markup. But we're still different from one another. We have different characteristics. We look a bit different. There are things that are unique to us. And that's the 0.1% remaining is what differentiates us from one another. That's the reason why we're not all look alike, talk alike, etc. So let's get back for a second. And after we spoke about those 23 pairs of chromosomes, let's explain a SNP. A SNP is a genetic markup. It's a specific location on a chromosome. All right? If you'll zoom in for a second on that area over there, it looks pretty much like that. Now, there are 23 pairs because we got one from dad and one from mom. And if we look inside, we can see that there are combinations. There are running letters on the chromosome, if you'd like. These are the genetic markups. Each one of those locations is a SNP. Is what defines us, is the value that we got. We have 4 letters, A, G, C, and T. And in each location, we can see a letter that we got from mom and a letter that we got from dad. We don't always know who we got it from. And on our entire genome, we have about 3 billion such base pairs. But as we mentioned, about 99.9% is similar between all of us. If we look at the 3 billion pairs of you and every one of you, we'll see that the A, G, C, T look exactly the same. That's why we identified as humans. So what we're really looking at is at that gap, that delta, the difference, those pretty much roughly 700,000 SNPs, which are unique to each one of us. So After we understood what a chromosome is and what a SNP is, a segment is pretty much a sequence of SNPs together and the results that they hold. So if I'll take the T and the G and the A from the 3 SNPs, we can pretty much call this a segment. This is a DNA segment with the values that I have in it, the results that I have, what I've inherited from my ancestors. So we know what a chromosome is, we know what a SNP is, and we know what a segment is. The remaining part is a centimorgan, which Diana also spoke about. And the centimorgan is the distance. We're using it to measure genetic distances between SNPs, between genetic markups on the genome. How much is it to walk from this SNP to the other? And that difference is what a centimorgan is, and it's a unit used in genetics to measure the distance between those SNPs, as I mentioned. If we'll go a little bit in depth and explain a little bit more how is it determined and what the value actually represents, it's the probability that a recombination event will occur between the SNPs in a generation every time a new child is born. And in the next slide, I'll explain what a recombination is and how does it look. So here are Frank and Molly. I've been using them for 2 years. They never disappoint me, and we'll keep using them. So let's look at Frank and Molly's chromosome 1, for example. Okay, so each one of them has his own genetic makeup, and Frank is represented in purple, and Molly is represented in orange. As I mentioned, these are pairs, one from dad, one from mom, so that's the reason why there are 2. And the day came, and Frank and Molly had a son. They had Ben, and this is how Ben's chromosome number 1 looks like. Okay? So I hope you can see that. Ben has one chromosome, one of the pairs colored in purple, and the other one colored in orange, because the upper one he inherited from his dad Frank, and the one on the bottom he inherited from Molly. And you can see that the colors don't look exactly the same because he inherited some from his father's first allele, we call it, and from his second allele. And let's look at it for a second and zoom in. So this was Frank's chromosome 1, and this is Ben's chromosome 1. And you can see that from the beginning till about 1/3 from here, it looks like, it's the more darker purple color, because he inherited that part of the chromosome from dad, which represents actually a grandmother or a grandfather, right? These are the 2 that Frank has. And then there was a recombination event. It switched. It stopped pulling DNA data from the first grandparent and then started taking the rest of the DNA from the second grandparent. And Ben ended up with having chromosome 1, which is a little bit from here and a little bit from there, with one recombination event. That's exactly that point. Molly contributed also DNA from her parents to Ben. And here we have 2 recombination events. So we started with one grandparent, stopped, moved to the other one, and then went back to the first grandparent, and she inherited these pieces of DNA from him. And this is how DNA is inherited in general. So we know that every generation we pass down about 50% of our DNA to our descendants, but these are not always the same 50%, and they look exactly the same on how they are distributed. Otherwise, me and my brother, for example, would have looked exactly the same and had 100% shared DNA if it was exactly the same recombination events again and again and again. That can only happen once, and that's in case of identical twins. For identical twins, they share 100% of their DNA, which makes life much more complicated when it comes to genetic genealogy. All right. So now let's talk a little bit about the science of DNA testing, okay? And how do we do that? Once we understood kind of the basic terms. So you got online, you got on MyHeritage, and you've ordered your DNA kit, and you're happy, and you've waited, and it came in the mail, and you've taken the test, and you've activated it online, and you sent it to us, and the lab processed it. And that's it. Your DNA results are ready after 3 weeks. I know that's the toughest part of waiting those 3 to 4 weeks. And that's it. They're ready. You're ready to see your results. Here they are. These are your results. This is your DNA. You can see the beautiful ethnicities that we were able to figure out, all your relatives that are here. So what we actually see here is that we see a very long list of SNPs and what you hold in them. What is the result? What are the 4-letter combinations that you have in your DNA, which based on them will be able to provide you with your results, with your ethnicity estimate and DNA matches. And there are about 700,000 SNPs in that file, which you can also download and use for various purposes. Now let's get back for a second and zoom in and see what we have here. All right? So the first column, which is named by RSID, is the SNP. That specific one location on one of the 22 autosomal chromosomes we use for DNA matching and ethnicity estimate. So you can see the name of that specific SNP, on which chromosome it is. This list of 700,000 goes from 1 to 22. The position, which is less relevant for this discussion, but it pretty much says where on chromosome 1 this SNP is located in the specific build that your array was made on. And most important, what do you have there? What are the letters? What are the value? What was passed down to you from your parents? So here we can see a C and a T, which I know that one of the parents contributed a C, one of the parents, the other parent contributed a T, but I can't know which one provided what. For that, we have something else, which I'll explain now. So Diane also mentioned that in her talk, there are a lot of things that we're doing from the moment that we get your DNA test results, the raw DNA data file, till you actually get your results online. And I'll talk more explicitly about 2 of them today, and that's on the phasing and imputation, because these are the coolest. But there's a lot more done until you eventually see your DNA results online. So phasing is our attempt to take your raw DNA data and break it down and say, this came from dad, this came from one parent, I stand corrected, this came from one parent, and this came from another one. So we need sequences, we need those segments separated, right? Because we can't really do DNA matching or provide you with an ethnicity estimate if everything is mixed up. We need to take those 2 letters that you see in each one of the SNPs and break them down into 2 lists, what came from parent A and from parent B. And that's the process in which, that's the process that is called phasing, in which we take those combinations of letters and we break them down into 2. Sometimes we won't be able to say which letter came from whom, because you have a TT, for example, so we know for sure that they both contributed the T. But there are some cases like if both parents had AB, AB, and you also have AB, we wouldn't be able to know, in this example we're using ABs, which one contributed it. But there are algorithms that we use internally that help us when we look at the broader picture, when we zoom out and not necessarily look at each SNP separately. When we look at the SNPs surrounding that SNP, we are able to do phasing much better. So once we were able to break them down into 2 lists, we move to the second step, which we call imputation. This is needed for 2 major reasons. The first one is that sometimes when we take a test, the lab can't figure out what is the result that you have in a specific SNP. Either the DNA that was provided in a malformed way, or the lab just failed with this specific test. And if we're going to have a missing piece there, it's going to be much more complicated for us to say, this is your ethnicity, or here are your matches. So we're doing imputation. We're trying to fill in the gaps. Another reason we're doing that is because we also support uploads from other companies, as you may know, and every company is looking at a different array of SNPs. It's pretty much 700,000 SNPs for all companies, but there's a difference between which specific SNPs each company looks into. So we need one reference. We need one file that we can compare one file to the other, apples to apples. We can't have those gaps. In the example here on the left, you can see DNA results, raw DNA data from a user who took a test with the MyHeritage chip, and on the right, someone who took a test with another company and uploaded the data to us. All right? So you can see that some of the SNPs appear both here and there, but some SNPs are unique to MyHeritage, and some SNPs are unique to the other company. And we need a common ground. We need to compare that. And the way we do it is by doing imputation. The best way to explain imputation is using this example. Can you read this? Now, you were, even though many letters are missing. The way it worked is that you looked at, again, the surrounding areas around where the letters are missing, right? So it's fine that I was missing in the word brain. Your brain was able to fill in the gaps and understand that I is the letter that is missing here. And this is exactly what we're doing with imputation. We're looking at the SNP surrounding, and we're making an assumption, a guess, what should have been there due to the other letters around. Now, for some, it may sound like this is like not fully accurate or things like that, and there are imputation errors, but it's way less than what you would have expected, especially when you're talking about 700,000 SNPs and you have a good reference of about 1,000 and one is missing. That's pretty easy to fill in the gaps. And this is exactly what we do. And once we run our algorithms, we can see that we can now match the file that I have from the MyHeritage chip and a file that I have from another user who uploaded. And now I can look and try to find those common and those shared segments, those repetitions that I can see in my file and in someone else's file. All right? So we spoke about phasing and we spoke about imputation. And now let's talk about how we're doing ethnicity estimate. So the definition I like the most for ethnicity estimate is the architecture of genome variation between populations. We know that various populations around the world have certain characteristics in their DNA that are unique to them. The tough part is to figure that out. First, define those populations, and then define what's unique to them. Sometimes it's done the other way around. We first find something that's unique to a group of people, and when we look at the people, we can see locations that come back again and again, or something that's unique to them. Now let's see how the inheritance of such ethnicities work. So we're back with Mike, and we're looking at his maternal side. Now, when it comes to talking about ethnicity estimates, to make everyone in the room on the same page and no political too much involved, we use colors. No one has anything against the purple color, I hope, or some strong feelings about it. So we can tell that Mike's great-grandparents, all of them were purplish. They come from the purple ethnicity, and it's unique to them. They are not mixed, which is also one of the challenges of defining who is unique, who is a founder, who is for that ethnicity. So let's assume all of them were purple. So that means that when they had their kids were also purple, and therefore our Mike is 50% purple. And that's from the maternal side of the family. When we're looking at the paternal side, things are a bit more complex. And here we have 3 new colors added to the equation. We can see that on the maternal grandmother side here, they are orange. And on the paternal, they're all red and blue. So when the DNA was inherited, and again, we keep about 50% of our DNA every time a child is born, we can see that Mike's grandparent on the paternal side is mixed. He's the first one so far that we've seen that he's mixed. He's 50% blue and 50% red because his grandparents coming from different colors, ethnicities. And when we're talking about his father, we can see that now his father is mixed with 3 ethnicities. And when we're coming down to Mike eventually to look at his ethnicity combination, at his ethnicity mix-up, we can see that after he's 50% purple, as we saw, all coming from his maternal side, he has 3 additional ethnicities at 25% orange and 12.5 both blue and red, because that's what his ancestors have passed down to him. But colors are nice, and eventually we do need to give them some names, and this is how we understand our history. So one thing that MyHeritage has done when we just started with the DNA testing about 2.5 years ago, almost 3 now, is that we kicked off a founder populations project. we had a huge database of people with very rich trees where they documented their past and where their ancestors came from. And we used that as reference to identify people who have lived and have a long history in the same geographical place, surnames, all these kind of things took part in the game. And we were trying to identify those who we can call founder population for a specific group. And some of them are unique, so we went all around the world and we chose various people from different backgrounds. Gilad earlier today spoke about our tribal quest, so we have also taken DNA tests from these groups. Some of these ethnicities are extremely unique to MyHeritage. And we've identified those specific ethnicities using the founder population data. So kind of to summarize that, we found a group of people who we believe are part of one group. We've tested all of them. We took the data and we tried to look for unique characteristics that we couldn't see with other groups. So for example, in chromosome 2, we see a segment that always looks like this, and this is the repetition, it's always the same. So we identified that as a segment that is related to Ashkenazi Jewish, for example, or for Greek, okay? And this is how we built our models, 42 of them today, but as you heard earlier this morning, this is about to change very dramatically in the next couple of months, which is super exciting. So we collected the founder populations data, and we've built the models for different ethnic groups. When a user result comes in, we look at his 700,000 SNPs, we do the phasing, we do the imputation, and once we have that data file ready to work with, we run it against our algorithms, and we're trying to say, we find 50% of his DNA looks like Ashkenazi Jewish, and 20% looks like Italian, and 10% looks like Scandinavian. And we're always matching it against the reference population that we used for the initial model, which went through some changes over the years, but more to come. Once we finish with that, we aggregate all these comparison results to the percentage per ethnicity, and this is what is served online. This is what the user eventually gets in the form of a nice interface with music and things like that. You like the music? Okay. DNA matches is done a bit differently, but pretty much the same concepts. I love my Frank and Molly. So they're back. So Frank and Molly had kids, and we spoke about their chromosome 1, for example, and how it looks and how each one of the alleles came from one parent. And guess what? Ben has a sister, Lisa. So this is an example of a family of 4, and you can see that Ben and Lisa, although they are brothers, as we explained, their DNA doesn't look exactly the same. It's in between 37 to 50%, depends on the company that you tested with. We're doing it a bit differently when it comes to siblings. And you can see that Ben and Lisa's chromosome 1, there are some shared segments. You can see some resemblance in some of the parts, but it's not really full. If I'll take just the parts that are exactly the same, you can see them on the right. And this is the amount of shared DNA between Ben and Lisa on chromosome 1. All right? It's about 50%. Time goes by, and Ben and Lisa meet their partners, and new kids are born, Eric and Julia. They are the grandchildren of Frank and Molly, and you can see that new colors were added, right? This is coming from their spouses. The blue from Eric's wife or partner, and the green from Julia's partner. And that's a completely new color. Now let's try and look at how much of shared DNA Eric and Julia have in common. And you can see that now it's way lower, right? On one of the alleles, one of the pairs of the chromosome, they share none at all because they have different moms or dads who are not related to one another. And the shared DNA on the first allele, on the first pair of the chromosome, is again cut in half. So now they have even less. So what we're doing when we're getting that raw DNA data file is that we're matching it against all of the DNA data files that we have in our system. So when a kit comes in, we're running it against 2 million at one point of time, and then against 2.5 million and 3 million and so on. And we're trying to look for people who have a decent amount of shared DNA between them to determine that this is a potential DNA match, there is a potential common ancestor. What we're actually doing is that we're trying to look for those sequences, all right? Those same repetitive segments or sequences of SNPs that repeat. So here with person 1 and person A and person B, we can see that there's a repetitive pattern of DNA on one of the chromosomes, and we highlight that and we say this is a shared segment, and we say what is the centimorgan for that segment, and how many total segments there are, and what is the total amount of shared DNA, which determines the relationship between two DNA matches. Okay? When looking, the percentage or the amount of shared DNA, there's about the total centimorgans in the human genome that we test is roughly 7,000 centimorgans. And what we're looking at is that the total amount of shared DNA so that we can tell you how you might be related. What is the connection between you and the DNA match that we found for you? In some of the cases, in most of the cases, it's more than one possibility. For 50%, for example, it can be either a parent, a sibling, or a child, right? One generation down, 25%. Now there are grandfathers, uncle and aunt can potentially be 25%, have 25% of shared DNA, and mirror the same thing for the other side. 6.25, 12.5, sorry. We're now talking about great-grandparents or great-uncles, first cousins, 6.25, and as we go lower, the number of possibility goes up. So on the website, what we do is that we take some of the user profile details so that we can narrow down some of the estimates. So if I'm matched with someone who is younger than me by 20 years, she probably cannot be my mother, right? She might be my daughter. And we're also using the gender so that we can tell you whether it's a female or a male, and so on and so forth. And as we go further, more possibilities what the type of relationship can be. And this is all really nice, and you eventually end up with a big list of DNA matches, but this actually represents the amount of shared DNA that you have that originated from a common ancestor, right? So the end goal for all of this, for those who are just starting or learning a little bit more about that, is to figure out how are you related, not just say that you're 1.2% and that makes you fourth cousins. That's great to know, or fifth cousins. That's great to know, but what's really interesting is who is the common ancestor that you both originated from. So if on the website, for example, my daughter here has a shared DNA segment with a Jane Doe, and she has a certain amount of shared DNA with her, that doesn't tell me much. What I really need to do is to keep on my research and try to go up the tree and look for more hints using genealogy, trees data, records, trees, of course, to try and understand where that ACC combination, that sequence that repeated both on my side, on my daughter's side and hers, where is it coming from? And one of the first thing that people do is, because I've tested myself and I've tested my wife, is that I can see that Jane Doe is also matching me, and therefore it's coming from her paternal side, and it goes up to my maternal side, and so on and so forth, until I am able to find who is that common ancestor. And that is the way that we're doing that. This is the way how we're taking the raw DNA data file, and we try to provide you with an ethnicity estimate that is most accurate according to the reference population that we have built on MyHeritage, and match you with the DNA matches on the system. The great thing about DNA matches is that your list keeps growing all the time because more people test, so it's not a one-time thing. It's not like the results that you see today might change tomorrow and more people will be added and a strong match may appear. And that is pretty much it for the basics. So thank you very much. And if there are any questions, I'll be happy to take. Thank you. Yes. Thank you, Marianne. Hello, Charles.
You said that you separate out the father and the mother DNA strand DNA fairly early on in the process in order to work out the matches.
And ethnicity, correct.
Can you actually take that further and show the matches, which side they come?
So if everyone heard the question? No, all right. So Charles was asking, so I explained about the phasing in which we take the two letters, structure the chromosome and try to break it into two alleles to say this came from this came from mom, this came from dad, and whether we can do much more than that and also present it in the interface on the website. This one is from mom and this one is from dad, if I got it right. So phasing, what it does, I don't know if it's from mom or from dad. You can sometimes do that. Some of the companies are doing matches on the X chromosomes, and if you're a male, then you're lucky and you have a match on the X, you can say that it's probably coming from your maternal side. Not always, by the way. So we don't really know if it's mom or dad. We can say parent A, parent B. And sometimes we won't even know, like we phasing for chromosome 1 and chromosome 2 and chromosome 3 will be left with two alleles, which we don't know which one came from what side. What we can do on the interface, and that is planned to take place soon, is that if another relative, a known relative of yours have taken a DNA test, and from the tree we are able to say that he's on your maternal or paternal side, we'll be able to surface that on the website and say, you are matched with a Jane Doe, and we can see that a cousin on your paternal side is also matching Jane Doe. So based on that, we can assume or give you a hint that Jane Doe might be related to you on your paternal side. These are what shared DNA matches are usually used for. Okay. Questions? Yes.
I was talking in the presentation about pilots. Do you have any plans to implement some sort of do you have any plans to implement an algorithm that will deal with pilots? So I've got, I don't know how many people piling up on chromosome 15 on about 30 centimorgans, and it's clearly, there's no explanation for these. And when I look at the raw data, the SNP count is actually quite low for a lot of these segments. So it'd be really useful if you could actually implement something that would actually downweight this overmatching on those pile-up segments.
So the question was regarding pile-up areas that Diane spoke about in her presentation. When we look, for example, to explain what a pile-up area is for those who weren't here in Diane's talk. So we see that there's a shared DNA in that specific position on the chromosome, right? Pile-up is an area which we see a lot of repetitive, a lot of the DNA matches, a lot of people keep sharing segments in that specific location on that specific chromosome or chromosomes, and it keeps coming up again and again and again. So that's probably not an IBD, not an identify by descent. It didn't come from us, from an ancestor, but it's something that's 10,000, 20,000 years ago, kind of common to people, to all people from that ethnicity, and that obviously should be ruled out or excluded when we're calculating the total amount of shared DNA, because this doesn't help us progress much, and it also kind of drives the relationship estimate away, because we're overestimating how closely you are. We're taking into consideration shared DNA that is not really because of a common ancestor or a close recent common ancestor. And whether the question was whether we plan to improve our algorithms to rule out those pile-up areas. And the answer is that we're even doing that today, but we're not doing it good enough, I must agree. If we find only one segment with a DNA match, and that segment happen to fall on a shared, on an area which is known to be a pile-up area, we're throwing it away and we're not taking it in. If you have two segments or more, and one of them happens to be, we're still including them. The tricky part is that sometimes it's right. So yes, we are working on enhancing the algorithm so that we can somehow find those, how a shared segments can be identified as because of a pileup or IBS or IBD. All right? Yes. Mariana is taking control.
Have you gotten any experience on mitochondrial DNA?
The question was whether I or MyHeritage have any experience with mitochondrial DNA. What I spoke about here was MyHeritage is doing only autosomal testing analysis on the 22 chromosomes. There are additional two known tests in the market. One is the Y, which males can do, and mitochondrial, which is following the DNA inherited from the maternal side. There are companies who do that, but on MyHeritage we are currently not, and there are no near-term plans to do mitochondrial or Y testing, for that matter. Yes.
I have a question about identical twins. I have two sons who are identical twins. They both have children. One has one son, and the other has two sons. What I wondered was why these two sons, different, they are cousins, don't look alike anything. They just don't look alike. They are half brothers. Genetically seen, these two sons.
I don't know them personally. I take your word for it.
You do understand my question?
I think I did. The question was, there are your sons, the identical
They're my
twins?
sons,
Okay.
and they're
She has two sons who are identical twins, and she has grandchildren from both of them, but they don't look any at all alike. Very possible. That's true for every siblings. I have a brother who doesn't look any way like me. The fact that they are identical twins, there's also another part of the equation that contributes about 50% to how they look. And that's, I guess, my best explanation for that. Obviously, and they also, what actually I can elaborate on more, as we explained, they didn't pass on the same exact DNA to their kids, your two sons. Because we're not every time both, so they're half brothers, that's great. They didn't necessarily, I don't know, like, contributed the gene that defines their hair color as black or as redheads or something like that, so they can look different because each one is passing down different parts of the DNA. They will share 50%, the brothers, and then roughly 12.5% for the, sorry? Yes. So there are specific genes that, SNPs genes that contribute to how we look and our physical appearance. And they have passed down different parts of DNA. Each one contributed something else, and they turn out a bit differently. Remember, there are two letters, so one might have gotten one letter and the other one got another one. Thank you. Sure. Yes, we'll take one more, and then it's your break time.
Hello.
Hi.
I'm wondering if it's possible to have DNA smart matches, because when I put more people in my tree and they have matches on DNA, I don't see them. I just have to check my whole tree and all my matches if there are new people on the tree.
So if I got the question right, you're saying that when you're looking at your smart match, you would have liked to see an indication that a person you are smart matched with is in a tree with which
DNA
you have
matches.
a DNA match.
Yes.
So we started working on something very similar to that. So today, when you're looking at your DNA matches, one of the ways to filter your list, you're usually looking at list of, I don't know, depends on your ethnicity usually and where you are in the world, but tens of thousands of people sometimes. And it's really hard to decide where to start. My personal preference, and I'm talking about it in a different session, is filtering the list by various criterias. One of them, which I like the most, is by those with whom you have a smart match. A smart match, for those who don't know, is a person who exists in your tree, and we found another person with a similar name, birth dates, and things like that, that we believe are the same people in both trees, which is a great indication how you might be related, right? And you're talking about the other way around. When looking at smart matches, it will be great to tell me that I have a DNA match with someone in that tree. We started playing around with it, so it's in the plans. All right. Thank you all very much. If you have any questions, I'm around here. I'm here for the next 2 days. So thank you. Enjoy the rest.